Hello and welcome to this Introduction to Microbiology video. I'm Dr. Aled Roberts, and in this video we're going to look at the bacterial cell envelope, its varying structures, their functions, and how we can use it as a very general tool to group bacterial cells. And so generally speaking, bacterial cells can be classified into two broad groups, and this is based upon key differences in their cell envelope, which is the structure that surrounds and defines the cell. And these differences are identified using a microbiological method known as the Gram stain technique, developed by Hans Christian Gram in the 1880s. Now, this technique applies sequential stains and washes to bacterial cells, and depending on which stains are retained, it gives us an indication of the structure of the bacterial cell envelope. Now, the two groups of bacterial cells differentiated by the Gram stain technique are the gram-positive and the gram-negative bacteria. Now, the gram-positive bacteria have a cell envelope composed of a single cytoplasmic membrane and a thick layer of peptidoglycan which is interspersed with teichoic acids. The gram-negative bacteria, on the other hand, have a double membrane structure to their cell envelope along with a thin layer of peptidoglycan and the presence of lipopolysaccharides. Now, at this stage, these differentiators might not make much sense, but we'll go through each of these components in turn over the course of this video, starting with our cytoplasmic membrane. Now, the cytoplasmic membrane of bacterial cells has the same basic phospholipid bilayer that we see in all cells. It has two layers or leaflets of phospholipid molecules. Now, each phospholipid has a hydrophilic head group that faces the aqueous environment and a hydrophobic fatty acid tail which faces away from the aqueous environment. And so together they form the membrane, which is about 7 to 8 nanometers across and is commonly described as having a fluid mosaic structure. This essentially means the phospholipids can move laterally within the membrane, giving it great flexibility whilst playing into its overall function, which is to effectively segregate the uncontrollable external environment from the controllable internal cytoplasm of the cell. And as we can see here in this cross-section, this is the boundary of our cell. Now, within the sea of phospholipids we have thousands of proteins, however they can be classified into some basic groups. First, we have the integral or intrinsic proteins, which span from the outer leaflet to the inner leaflet. And they come in a range of forms. We have transporter proteins, which act as a gateway, allowing materials to move into and out of the cell, passing from one side to another. We can also have receptor proteins, which interact with materials in the external environment and pass a signal into the cytoplasm without anything physically moving into or out of the cell. Then we have our second group of proteins, which are the peripheral or extrinsic proteins, and they associate with only one leaflet of the cytoplasmic membrane. Essentially, they provide alternative functions such as the enzymatic degradation of waste products or the metabolism of complex materials so that they can be more readily transported into the cell via the transporter proteins. Okay, so apart from defining the cell, what is the overall function? Well, it actually has three key functions. The first is that it acts as a permeability barrier to most substances, especially polar or charged molecules. Now, as a result of this, transport proteins are essential for moving materials in and out of the cell, sometimes against concentration gradients. The second is that it provides a space for proteins to anchor to. Now remember, this anchoring is just within the membrane. Due to the fluid mosaic nature of the membrane, these proteins move laterally around the membrane. And finally, it allows energy conservation and consumption, which essentially means it can be used to generate energy. And it does this by pumping protons across the membrane. The cell therefore creates a charge and pH difference known as the proton motive force, which is a form of stored energy, much like a charged battery. Now, this electrochemical gradient can then drive energy-dependent processes such as nutrient uptake, flagellar rotation, and the synthesis of ATP. Now, whilst the cytoplasmic membrane is relatively fluid, surrounding it is a much more rigid structure known as peptidoglycan. Now, peptidoglycan is a rigid mesh polymer of long-chain carbohydrates that are linked together through short peptide chains, which is quite a lot to process, so let's break this down. First is the sugar backbone. Now, peptidoglycan contains thousands of long sugar chains which are formed from two very specific sugars, N-acetylmuramic acid and N-acetylglucosamine. Now, these two sugars alternate with one another along the chain and are joined together via beta-1,4 glycosidic bonds. And if we were to zoom in, we can see here the rough structure of N-acetylmuramic acid on the left and N-acetylglucosamine on the right. Now, both sugars are based upon a slightly modified glucose molecule, so a 6-carbon hexose sugar. At carbon number 2, the hydroxyl group we might expect in glucose is replaced by an amino group which becomes acetylated, forming the N-acetyl. Now, as you can see from the diagram, they differ significantly on carbon number 3, where we have a lactic acid group attached via an ester linkage onto the N-acetylmuramic acid sugar. And what this does is provide a reactive site to help form crosslinks. More on this in a minute. Now, as I said earlier, these two alternating sugars are linked together via a beta-1,4 glycosidic bond. So carbon 1 of one sugar covalently binds to carbon 4 of the next in such a way that it forms a straight chain. And so what we end up with is long chains of alternating sugars, which can be hundreds if not thousands of residues long. Now, if we remember back to our N-acetylmuramic acid, it has a reactive site, and it is this site that bacteria use to form short peptide chains. Now, these chains are typically 3 to 5 amino acids in length, depending on the species, and they contain some quite unique amino acids that we don't tend to see too often. And so if we zoom in, we can see the 5 amino acids observed in Escherichia coli. Now, these are added sequentially to carbon 3 of our N-acetylmuramic acid, forming a pentapeptide. Now, the amino acids which form this pentapeptide are L-alanine, D-glutamic acid, diaminopimelic acid, D-alanine, and another D-alanine residue for good measure. Now, what makes these amino acids somewhat unique is the presence of D-form amino acids. L forms usually find their way into proteins in nature. However, these D forms give a unique structure and make it more resistant to degradation by host enzymes. In addition to this, we have the presence of mesodiaminopimelic acid, which is probably an amino acid you haven't heard of before, mainly because it's not found in humans. Now, what makes this even more interesting is that it has a free amide and carboxyl groups, which can act as reactive sites, and again, more on this in a minute. And I guess finally, it's quite common for more amino acids to be present during the building stage. So when forming this short side chain in E. coli, 5 amino acids are added. However, when peptidoglycan is formed, the terminal D-alanine residue is lost as a crosslink forms. But what actually does this look like? Well, if we bring back our peptidoglycan chain complete with peptide side chains and add a few more chains, they can start to orientate next to one another, allowing some of the pentapeptide side chains to come into close contact with one another and form what we call a crosslink. Now, there are two types of crosslink. We have direct peptide crosslinks and interbridge crosslinks. Now, each bacterial species can form just one of these. So for instance, Escherichia coli forms direct peptide crosslinks, while Staphylococcus aureus forms interbridge crosslinks. But how do they differ? Well, the direct peptide crosslink is sort of exactly what it says on the tin. The amino acid side chains covalently bind to each other directly. Interbridge crosslinks, on the other hand, are a little more complex. This is where a short peptide bridge is added between the two side chains. This is usually around 5 amino acids, and in the case of Staphylococcus aureus that I just mentioned, this bridge is made of 5 glycine residues, known as a glycine bridge. Now, the location of these crosslinks is usually between the diaminopymeric acid of one side chain and the D-alanine of another. Now, the purpose of these crosslinks is to join together multiple strands of peptidoglycan, increasing the rigidity of the structure. Now, when we look at peptidoglycan, it's important to remember that it is a three-dimensional structure. Along the x-axis, we have long chains of our alternating sugars, which are held together via beta-1,4-glycosidic bonds. Then along the y-axis, we have the short peptide chains that link the sugars together via peptide bonds. Now, something which isn't apparent at this point is layering. So what we can see now is a single layer of cross-linked peptidoglycan. However, in reality, we have multiple layers stacked up on top of one another like this. Now, these layers in the z-axis are also cross-linked through peptide bonds with peptide side chains in other layers. So that's the rigid mesh-like structure of peptidoglycan. But why do bacteria have peptidoglycan? Well, first and foremost, it provides structural support to the cell. So if you remember, the cytoplasmic membrane is quite fragile, whereas peptidoglycan is rigid and therefore provides some much needed structural stability. Now, this is particularly crucial for protecting the cell from osmotic stress, stopping bacterial cells from taking on too much water to the point of cell lysis, helping to limit excessive expansion. In addition to this, it plays a key role during cell division, acting as a scaffold for the construction of the new cell wall. It also serves as an anchor point for various proteins, ensuring they remain in place where they are needed. And finally, perhaps less of a function and more of a consequence, it can play an important role in how our immune system detects bacteria, and that is because fragments of peptidoglycan can be shed during growth and division, which then triggers inflammatory responses and allows our body to respond quickly to their presence. Now, sometimes, and we'll come on to when a little later, outside of the peptidoglycan is a second phospholipid bilayer known as the outer membrane. It has a similar basic construction to the cytoplasmic membrane and therefore has many of the same functions and characteristics, except when it is present, instead of segregating the cytoplasm from the external environment, it separates the external environment from a newly created area known as the periplasmic space. And so if we visualize this, we can see that the cytoplasmic membrane separates the cytoplasm from the external environment, which all cells have, and outside of this is the second outer membrane, which we can see here. And just as I said, it creates a new environment known as the periplasmic space, which houses enzymes that process nutrients, synthesize peptidoglycan, detoxify compounds before they reach the cytoplasm, and helps with environmental sensing. Now, the outer membrane, just like our cytoplasmic membrane, contains a number of important proteins, and so we have transporter proteins and receptor proteins, as well as various membrane-associated proteins. Now, in addition to all of this, the outer membrane houses specialized transport proteins known as porins. Now, porins have a homotrimeric protein structure, which aid the movement of water-soluble molecules from one side of the outer membrane to the other. Now, this is done via passive diffusion and therefore is an energy-independent process. However, as a result of this, there is a cut-off size limit for the molecules that can pass through, usually less than 700 Daltons, which will allow for things like nutrients, waste products and antimicrobial agents to move through them, but not larger molecules such as proteins. Now, there are many different types of porins found in the outer cell membrane, each with their own amino acid composition. Now, changes in this composition affects the types of molecules that can pass through, resulting in some very general porins that allow the movement of lots of different molecules, and some very specific porins that only allow very specific molecules to pass through. Now, embedded in the outer membrane is one final structure, lipopolysaccharides, which cover the entirety of the outer membrane and extend outwards into the external environment. Now, when we look at this molecule, it is essentially a complex glycolipid that has three distinct sections: the lipid A section, the core polysaccharide, and the O antigen polysaccharide. Now, the lipid A section is the innermost portion that anchors the LPS into the outer membrane. It's made up of a disaccharide backbone of glucosamine units with attached fatty acid chains. This region is highly conserved across gram-negative species because it's essential for maintaining membrane integrity, but it's also used to cause damage and disease within a target host. More on this in a minute. Next, we have the core polysaccharide, which connects lipid A to the O antigen, and as the name suggests, it is composed of multiple sugars. However, they aren't your standard sugars like glucose and fructose. They are heptoses and others known as 3-deoxy-D-mano-oct-2-ulosonic acid, thankfully abbreviated to KDO, and this is attached to phosphate groups. Now, this part shows genus level variability, meaning its structure tends to differ between bacterial genera but remains fairly consistent within a genus. It helps provide stability to the outer membrane and contributes to the overall negative charge of the cell surface. And finally, we have the O antigen, which extends outwards from the core as a long chain of repeating sugar units, with each sugar unit having between 3 and 5 sugars, which are repeated around 10 to 25 times on average. Now, this is the most variable region, differing not just between species, but even between strains of the same species. So, for instance, the O antigen composition is what determines serotypes in things like E. coli 0157 versus 055. So what exactly does the lipopolysaccharide do for the bacterial cell? Well, it's actually doing a few really important jobs at once. First, it acts as a permeability barrier. The tightly packed lipopolysaccharide molecules form the outer leaflet of the outer membrane and make it highly resistant to many harmful compounds, things like biosalts, detergents, and large antibiotics, meaning this barrier only allows small hydrophilic molecules to pass through porins, keeping out many toxic substances that could otherwise damage the cell. Second, lipopolysaccharides help to stabilize the outer membrane. The phosphate groups in lipid A and the core oligosaccharide interact with divalent cations such as magnesium and calcium, which forms ionic bridges between neighboring lipopolysaccharides. This cross-linking effect keeps the membrane structurally rigid and prevents it from falling apart under stress. And finally, lipopolysaccharides are recognized as a pathogen-associated molecular pattern, or PAMP, meaning it's a molecular signature that our immune system uses to detect infection. The lipid A portion, in particular, acts as an endotoxin, triggering strong immune responses such as inflammation and fever when it's detected by immune receptors like TLR4. So overall, lipopolysaccharides are not just a structural feature, it's a multifunctional molecule that protects the cell, holds the cell envelope together, and plays a major role in how bacteria interact with their hosts. And that concludes our look at the different basic structures of the bacterial cell envelope. Now, as I said earlier, there are two different forms the bacterial cell envelope typically takes. We have our gram-positive bacteria and our gram-negative bacteria, which, based on the descriptors we can see on the screen right now, hopefully make a little more sense. But if we were to summarize this, our gram-positive cell wall has a single cytoplasmic membrane which encloses the cytoplasm and a relatively thick layer of peptidoglycan outside of this. But what do we mean by relatively thick? Well, it will typically have anywhere between 15 and 80 layers of peptidoglycan stacked on top of one another, which gives a total thickness of between 20 and 35 nanometers, meaning it will typically make up between 60 and 90% of the gram-positive cell envelope. Now, interspersed between peptidoglycan sheets are teichoic acids, which are polymer chains of glycerol or ribitol phosphates covalently linked to different peptidoglycan layers. This helps to provide structural stability whilst also helping to regulate ion movement through the cell wall, contributing to surface charge and influencing how cells associate with different surfaces. In addition to this, we also find lipoteichoic acids, which are structurally similar to our teichoic acids but anchored into the cytoplasmic membrane via a lipid tail. And overall, lipoteichoic acids help to keep the peptidoglycan associated with the cell membrane and have some functions in cell growth and division. Then finally, the cytoplasmic membrane is littered with hundreds of different intrinsic and extrinsic proteins, facilitating the movement of molecules and signals into and out of the cell. And that sums up our gram-positive bacteria. Then, on the flip side, we have our gram-negative bacteria. So our gram-negatives have two membranes, an inner cytoplasmic membrane which contains the cytoplasm and an outer membrane that creates the periplasmic space, which a relatively thin layer of peptidoglycan is found. But how thin are we talking? Well, it's typically only 1 to 3 layers of peptidoglycan, occasionally a few more, giving a total thickness of roughly 2 to 7 nanometers, so it makes up only a small fraction of the gram-negative cell envelope, usually only 10%. Now, instead of teichoic acids, the outer membrane has long lipopolysaccharide chains, which create a strong permeability barrier, contribute a negative surface charge, help resist biosalts and many antibiotics, and is a key trigger for host immune recognition. In addition to this, we also find outer membrane lipoproteins, especially Braun's lipoprotein, which is anchored to the outer membrane by a lipid tail and covalently linked to the peptidoglycan layer. And overall, these lipoproteins tether the outer membrane to the cell wall, helping to maintain envelope integrity during growth and division, and they work alongside porins, general and specific porins, that form channels for hydrophilic nutrient uptake. Then finally, both the cytoplasmic membrane and the outer membrane are littered with hundreds of different intrinsic and extrinsic proteins, facilitating the movement of molecules and signals both into and out of the periplasm and cytoplasm. And with that, we come to the end of this video. Hopefully you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.